Welcome, everyone. Thanks. Welcome, everyone, to this month's seminar series for the Society of Functional Precision Medicine. Before I introduce today's speaker, just a couple of things. One is keep an eye on your emails over the next few months. Please set whatever blocking tools you have to accept SFPM mailings. We do intend to be doing a little more contacting and setting up some working groups to solve problems that we think the SFPM needs to solve. So for all of you, you can contact me directly if you have ideas for things that you think the SFPM should be doing. And this is a good time to hear from you because we are hopefully going to start to formulate the groups to address some of these things. I also want to let you know that we are planning for the AACR meeting in April at having a reception associated with the AACR meeting. Probably not a formal scientific meeting, but keep your eyes posted for notification about that. It'll be somewhere right next to the convention center is how we're planning right now. Without any further ado, I am very happy to present today's speaker, Don Ingber. Don is the founding director here at Harvard of the Wyss Institute for Biologically Inspired Engineering at Harvard University. Judah Folkman, professor of vascular biology in the Department of Surgery at Harvard Medical School and the vascular biology program at Boston Children's Hospital, and also the Hans-Jörg Wyss professor of bioinspired engineering at the Harvard John Paulson School of Engineering and Applied Sciences. He received pretty much all his degrees at Yale University. He's a member of the National Academy of Medicine, as well as several other national academies that I won't bother listing. As you might expect, as someone who's so translationally motivated, hundreds of patents, and he's been listed among the world's top 20 translational researchers in 2012, 2019, and 2020. He'll be talking, I think, today quite a bit about an innovation that I think will be very interesting to people here, which is what he refers to as his organ-on chips as a way of doing pretty elegant biological testing of biological materials. And I have to close off this introduction with something I don't think I've ever introduced anyone before, saying that actually one of his devices, the organ-on-chip technology, was acquired by the Museum of Modern Art in New York City for its permanent design collection in 2015, which is a pretty unique achievement for a scientist, I think. So Don, take it away. With regard to questions, you are welcome to either enter your questions in the chat and I'll put them to Don, or we'll have time at the end where we can do questions live. Please just wait, put your hand up, and please let me call on you for you to ask your question at that time. So Don, please take it away.
Great, thank you so much. I'm just trying to see if I can get a pointer here. 1 second. Oh, well, we will go without it. So what I'd like to do today is describe to you technology that you heard mentioned called human organ chips that we have developed for disease modeling, drug development, and personalized medicine. And as you heard, I'm the founding director of the Wyss Institute at Harvard. And we're about 14 years old. We were started with what was then the largest single philanthropic gift to Harvard of $125 million, which we've replicated many times since. But when we were given that gift, we were tasked with taking on really tough problems that could potentially be, that would be very high risk, but could be potentially transformative. And one of the biggest problems that I could see at that time was that the drug development model is really challenged, if not broken. It currently costs $3 billion or more to go from the lab bench to approval. And there are many reasons for this. Animal studies are required. They take years to complete. There may be major ethical issues, but the real problem is that 70% of the time on average, and in some areas over 90% of the time, including cancer, the results from animal studies don't predict clinical responses when you get into clinical trials. And so as a result, there's been a search for alternative methods that could help one develop drugs that are safer and more effective, but much more quickly and at a lower cost. And so over the past 15 years or so we're working in the field, we also realized there are other challenges, which is that there are currently 40% of the drugs in the development pipeline are biologics, The FDA usually requires non-human primate studies. Obviously, non-human primates have ethical concerns, but they're in a terribly short supply. But the real problem is that many of these biologics are getting so specific for human targets. Think of CRISPR RNAs and RNA therapeutics, that they don't cross-react with non-human primates. In fact, oftentimes with monoclonals, they have to make a separate composition of matter that recognizes a different species, and then they move a human version into clinical trials, and it's sort of crazy. So this is another desperate need for preclinical human models. And finally, we've gotten into the area of microbiome research, which, we, over the last 15 years, major paradigm shift that it's so important for health and disease. But the microbiome differs totally between different species, even between different individuals, and sometimes even in the same individual over time. So right now, there are no good test beds for human microbiome-based therapeutics, which are beginning to enter into the clinical arena. So when we started the Institute, we had 5 major platform areas, and one I headed myself that essentially was focused on developing what we now call human organs on chips. And the idea was to use computer microchip manufacturing technology, which gives you control of features at the same nanometer to micrometer scale that living cells and tissues live at. To use this to create microchips containing living human cells that reconstitute organ level functions, not cell or tissue, but organ level functions, to accelerate drug development, replace animal testing, and advance personalized medicine. Now, our first success is what we call the human breathing lung on a chip, published in Science over 12 years ago. And we're not trying to build a whole organ. I like to think of these as living 3-dimensional cross sections through a major functional unit of an organ. And this first paper, we were developing a model of the alveolus, or the air sac. And for those of you who are not totally familiar, the alveolus is a relatively simple structure. I'm just trying to see if I get a point here once again. Yeah, there we go. It is filled with air. If you go to a high electron microscopic level, there's air. It's lined by a single layer of epithelial cell layer, the alveolar epithelial cell, then a porous, flexible extracellular matrix, the basement membrane, and then a capillary cell, and then blood. What this doesn't show you is that it's an incredibly mechanically active structure. Every time you breathe in and out, all of these structures stretch and relax. And my own work for 45 years, really, the core of what I've done is this idea that mechanical forces can be as important as chemicals and genes. And so this is something I felt is critical to incorporate into any model. And of course, respiratory physiologists know it's absolutely critical for lung function and lung development. And so if you were to try to distill down, like, what are the simplest design principles that behind an organ, for us, it was, we had to recreate this tissue-tissue interface, because that's what makes an organ. 2 or more tissues that interface and new functions emerge, of which usually one is a vascular tissue. And in the lung, we wanted this dynamic breathing motions, and we also wanted fluid flow and airflow. So this next animation shows you how this works. The top right, the chip is the size of a computer memory stick. It's made out of flexible silicon rubber, optically clear. If you cut it in cross section, it has 3 hollow channels, each less than 1 millimeter wide. The middle channel is cut into top and bottom by another flexible rubber membrane with pores that we coat with extracellular matrix. To make the lung chip, we take lung alveolar cells, put them on top, human lung capillary cells, put them on the bottom. We just recreated the alveolar capillary interface. And then the trick is we apply cyclic suction through the side vacuum chambers. It's flexible, so it stretches and relaxes at the same rate and degree when you breathe in and out. We then could put air over to create an air-liquid interface on the top chamber. We could flow medium with the immune cells, or even whole blood for short times if we put capillary cells on all 4 sides. Now, if this were to work, it should mimic an organ level response. So imagine you have a bacterial infection. What happens is there's a tissue-tissue signaling response. The epithelium put out cytokines that activate the endothelium to express receptors like ICAM. White blood cells that were just flowing by now stick, roll, diapedese, or migrate across and engulf the bacterium, and that's homeostasis. Now I'm going to show you imaging of the device. These are fresh white blood cells. I know we're fresh. We took them out of my postdoc. You only see the cells, the endothelium is unlabeled, and then the epithelium is behind the screen. To begin with, it's a healthy vessel, they just flow by. Now we put bacteria on the other side. Now there's that signaling response, ICAM is expressed. Now you're watching the white blood cells being pulled out under flow, which is important because the initial adhesion step is shear stress dependent. Now we can do any imaging on these devices. So here's higher magnification. This is one white blood cell right about there. It finds a space between two endothelium, goes beneath them, goes through the matrix-filled pore, wiggles its rear end out of focus here. Now you see it coming out the other side by phase contrast. And now I'm going to show you the white blood cells in red because the bacteria are labeled with GFP in green, and you see them being engulfed. So you just watch the entire human inflammatory response at high resolution in this little rubber chip. Now, at the time, I was not funded to do this. I was funded to work on nanotoxicology, but one of the major sites where nanoparticles enter the body is the lung. And so we essentially did studies where we used simulants of smog air pollutants, silica nanoparticles, and we put them into the airspace. And we used microfluorimetry to measure injury in terms of oxygen-free radical production fluorescently. And what you see in the graph is quite interesting, is that if we see injury if we have the full breathing motions on this chip, but we do not see any effects if it's static, as if a transwall culture, for example. And with this injury, we see recruitment of immune cells and inflammation. Now, we then actually looked at absorption of these particles, and we found that only with breathing is there major absorption of these particles, and there was an 8 to 10-fold increase, and there was no change in transport or absorption of fluorescent albumin or dextran. So this actually turned out to be mechanically induced transcytosis response across two cell layers, which may very well be mimicking how viruses get into the body. Now, this was not mimicking in vivo. No one's ever seen this before. So we developed an ex vivo ventilation perfusion model in a mouse, used the nebulizer to introduce the same particles while either having breathing motions or no breathing motions. And we got exactly the same response. And so this was a full article in Science. And I think this, we were told by FDA and NIH that this showed that this is possible and opened up the entire field, which is now a multi-billion-dollar industry. Now, early on, we started to talk to pharmaceutical companies, and they said, well, this is interesting, but we're more interested in toxicity models and particularly disease models, which is where animal models fail so badly. So we hit two birds with one stone by developing a model of pulmonary edema, fluid on the lungs. And we did this by using the FDA-approved cancer drug interleukin-2, which has a dose-limiting toxicity of pulmonary vascular leakage leading to pulmonary edema. So at time zero, this is air above the chip, looking from above what it looks like. And now we perfuse IL-2 at the clinical Cmax, and what we see is liquid filling the airspace like a meniscus of fluid over 2 to 4 days, completely filling it by day 4. And this is exactly the same time course as seen in patients. And we can quantify this using a trick from kidney physiology, fluorescent inulin. And what we see is, once again, minimal changes when the chips are static, but significant changes, as I just showed you, when there's physiological breathing motions. Again, we went back into animals and we confirmed this has never been seen before, but breathing actually has a major effect on this toxicity response. Now, this is important because clinically, most of these patients with pulmonary edema are on a ventilator, and you could actually impact the mechanical motions there. But as I mentioned, I've been interested in mechanotransduction for 40 years, and we had shown a few years before that one of the fastest mechanical signaling events when you stretch cells adherent to a flexible matrix is activation of an ion channel called TRPV4 that induces calcium influx, and that's upstream of integrin signaling and tyrosine kinases. And I knew that GlaxoSmithKline was working on an inhibitor of that channel, and I heard the program wasn't going so well, and I asked would they give us drug to test, which they did, and we completely prevented pulmonary edema in this human model. And they then did studies in parallel in dogs and rabbits with cardiogenic pulmonary edema, a physical cause of back pressure caused pulmonary edema, and they got the same results. And we had back-to-back papers in Science Translation Medicine, and that drug is now in phase 2 clinical trials. Now, what was interesting is that one reviewer said, this should not be published because it's too simple. There's no immune cells here. The other reviewer said, this is kind of amazing. It's synthetic biology at the cell, tissue, and organ level. They just showed you don't need immune cells for pulmonary edema induced by interleukin-2. And this is something we've seen again and again and again. We can control every variable, the cell types, the medium, gradients of oxygen, mechanical motions independently. And we could actually get insight into mechanistic control parameters that are impossible to do in animal models or human. So this one model, this lung alveolus chip, provided proof of principle for a disease model. We also used these chips to show drug toxicities of a drug that caused deaths in phase 1 clinical trials due to pulmonary thrombosis. We saw that on chips, but you couldn't see it in animals. Drug efficacy, therapeutic target discovery, new drug discovery. We've also done adenoviral vector delivery on these chips. And it's won multiple awards. You heard it's in the Museum of Modern Art. It's also won it beat out the Google car and a Frank Gehry building for the International Design Award a number of years ago. We've integrated immune cells, as I showed you in the movie, but we also have integrated stromal fibroblasts for pulmonary fibrosis studies. And again, you can build complexity as you desire. Now, we then we talked to other pharma companies who basically said, well, we're interested in lung as a target for therapeutics, but we're more interested in diseases of the small airway, like chronic obstructive pulmonary disease, COPD, or asthma. So we then developed a model of the airway. We got airway epithelial cells on top, air-liquid interface, lung endothelium on the bottom. Keep them at an air-liquid interface to differentiate for 3 weeks. And what you can see here is you get a beautiful ciliated epithelium. The cilia move in a directional way. If you put fluorescent microparticles at the bottom right, this is mucociliary clearance at the same rate on the chip as it is in all of our lungs at this very moment. And then we made chips with cells from patients with disease, in this case, COPD. And what we see after 3 weeks at an air-liquid interface, only feeding through the vascular channel with medium, no immune cells, they retain the COPD phenotype as indicated by decreased expression of TOL-R3 and 4 receptors. And now what brings COPD patients to the emergency room are exacerbations, either by bacterial infection, viral infection, or cigarette smoke. And so in these early studies, we mimic viral infection with poly-IC or bacterial infection with LPS endotoxin. And we could see that healthy chips didn't notice it, but COPD chips increased expression of various cytokines like IL-8 and MCCSF. And then we built a cigarette smoking robot. This is a cigarette lighter from a car. This is a Gatling gun. And this actually creates real smoking, mimic smoking parameters in terms of pulse intensity, pauses. And with this, real smoke goes to the airspace of the chip, not extract. And this is just 2 examples. If you look at the major inflammatory cytokine IL-8, smoke didn't do anything to a normal chip, but a chip lined by cells from COPD patients doubled the IL-8 levels. And at the right, you see transcriptomic analysis of 3 chips, as shown at the right, compared to 9 patients from a past study, where the chips are the change in gene expression after exposure to cigarette smoke, and the 9 patients are changes in gene expression that are observed in patients who have a history of cigarette smoking without any known obvious disease process. And we were very excited. The bottom 3 quarters to the top quarter were pretty similar, but the reviewers had a major problem that our chips were more similar to each other and there's greater variability in the real patients. And then we realized that in a sense, we could do this type of clinical study better than a clinical study, because this is matched comparative modeling of the same patients before and after the stimulus we're interested in, smoke exposure. And these genes are indeed related to smoking. Whereas these patients, obviously many are, but many others are related to work history, family environment, other disease process, et cetera. And this eventually got in, and we're asked to do a little mini review on using this for matched comparative modeling. More recently, Cystic Fibrosis Foundation reached out, gave us cells from patients with cystic fibrosis, put them on the chips, and as you see at the top, they have many more cilia, as shown in the top right, across the long channel of the chip, which is what's seen in patients. The beating frequency in the CF chips are higher, whereas you don't see this in trans wells that are static, but that's what it is in patients. There's more mucus production. There's higher baseline inflammatory state, as in patients. And you have higher cytokine production of IL-8 and lower IP-10. And we can begin to study microbiome. This is Pseudomonas aeruginosa on the chip, which is in all of our lungs, but is a common cause of morbidity and mortality in cystic fibrosis patients. And as you can see, rampant overgrowth of this bacteria on chip. Now, basically, what we realize from these sorts of studies is that these organ chips provide a window on molecular scale activities inside living human cells within a relevant tissue and organ context. And thus, this could enable new mechanistic insights and even drug discovery, plus provide a way to experimentally test what we discover or what one is considering for use in a human relevant model. So here's an example in terms of cancer. I've worked on cancer since I'm 19 years old, and in the 70s and 80s, the big advance was developing orthotopic cancer models, where rather than doing a sub-Q tumor in a mouse, you do a mammary tumor in a mammary fat pad or a prostate tumor in the prostate gland, and it looked a little bit more like in vivo. So we had these organ-specific chips, so we said, let's do human orthotopic cancer models. So we used the H1975 cell line, which comes from a human non-small cell lung cancer, but an adenocarcinoma form of the disease that clinically emerges in the distant bronchiole, the small airway, but preferentially grows in the alveoli. So we had airway chips and alveolus chips, and so we started by plating the cancer cells at a very low density relative to the normal cells on the upper channel with endothelium in the bottom. And what we found is that the top is the small airway chip, the bottom is the alveolus chip. So the first thing is the gray circles are the rate at which these tumor cells grow in a plastic dish with serum-containing medium as they've been cultured for 50 years, very rapidly. The white circles are the growth on a plastic dish of the same tumor cells in the medium we use for perfusing our chips through the vascular channel. They do not grow. So all the growth I'm going to show you is due to the microenvironment of the human lung cells. Okay, so in the small airway chip, they don't grow for 10 days, and then they grow slowly. Whereas in the alveolus chip, they grow rampantly from the beginning. And I should note that the airway chip has a thickened layer, so we can actually go through with a needle and inject tumor cells after the tissue is differentiated. When we do that, we just get a dormant tumor in the small airway. Now, we then did our experiment with or without breathing motions, and we found that breathing suppressed tumor growth in the lung alveolus chip by 50%. Now, this is also a very nice model to look at invasion. If you use GFP-labeled tumor cells, you can watch them going through the pores of the membrane and quantify it, and we saw that breathing inhibited invasion by 50%. And we actually were able to show, using a first-line drug like ruxolitinib, which is a tyrosine kinase inhibitor, that without breathing, where the tumor is growing rampantly, you get suppression of growth. But on the chip where it grows slowly, it grows right through drug treatment. And we were able to show in this paper that actually breathing regulates EGFR receptor tyrosine phosphorylation, which is what this drug is in part targeting. And so this, again, gives you new insight into not only mechanism of disease, but mechanism of drug action or drug resistance. Now, about this time, DARPA and NIH, 2016-17 came to us and said, could you leverage your lung chips to develop models of potential pandemic respiratory virus infections? And the focus on that time was influenza. And we did this, and here's GFP H1N1 influenza watching in real time. And you could beautifully infect these chips. In this slide, you can see that viral infection, GFP virus, is taken up by the cells. You lose cell-cell junctions, lose cilia. There's even endothelial damage. And when we use different strains of virus, H1N1, H3N2, H5N1, you see different levels of virulence that scale with what you see in patients. You could also measure compromised barrier function. But more interestingly is that you really can analyze host responses to infection as well as drugs. So what you see at the top is basically a cytokine storm that we're all familiar with from COVID, but for influenza, where you see multiple different cytokines increase in response to infection by viruses, and different strains give you different levels of cytokine production, which again scales with exactly the differences that are seen in patients. We can not only quantify immune cell recruitment, as I showed you earlier in the movie, but you even see clearance of infected cells. And now we could start using drugs, and this is oseltamivir is Tamiflu, the first-line influenza drug. And when we use it at the Cmax, as seen in patients, you get beautiful inhibition of viral infection. But more interestingly, that drug is only approved by the FDA to use in the first 48 hours after infection. And that's exactly the time window we see on these chips. Even more interestingly, we did an experiment where we infected a chip and treated with a drug at 90% efficacy. And then we trying to model human-to-human transmission of virus to see if we can get evolution of spontaneous variants of concern, if you like. And we basically cough a droplet or move a droplet from one airspace to the other while continuing to treat with the drug at 90% efficacy. When we did amantadine in 8 passages, we got virus that was completely resistant to drug. When we did genome sequencing, we found all of the known mutations that have been published in the literature of patients who spontaneously became resistant to amantadine. And we discovered a few new ones that no one's ever seen before. So you might be able to use this to predict what variants are going to emerge over time, or potentially do this with cancer cells as well. I should also note, when we did it with Tamiflu, it took 25 passages, but we got resistant virus, which is very, very scary. And this is when COVID-19 hit in January 2020. And my 2 new postdocs working on the viral infection model were following this on social media in China. And the day after the science paper came out in January, mid-January, with the SARS-CoV-2 gene sequence, they engineered SARS-CoV-2 pseudoviruses expressing the SARS-CoV-2 spike protein, because we only have a BSL-2 lab. And they did what, it was a desperate time. And so they did what they did in the graduate student labs, which is they used this cell line that is good for growing viruses, HUH7, probably because it has abnormal interferon response. And they used this pseudotype virus, and they went to the literature and they found 30 drugs that had previously been shown to have activity against related viruses like SARS-1 or MERS or Ebola. And we tested them in this cell line. And what you see here are the 8 drugs that showed dose-dependent inhibition in the low micromolar range. And this is, again, February 2020, chloroquine and hydroxychloroquine are in this list. But then we tested it on our lung chips, where we're using it at Cmax dose, flowing it through the vascular channel. Now hydroxychloroquine had no activity, nor did many of the other drugs that also didn't have activity in humans. And we found that a related antimalarial, amidiaquine, actually showed more significant activity. We were then funded by DARPA to collaborate with Ben Tenover in New York, who had a BSL-3 lab. He tested it against native SARS-CoV-2. We showed significant inhibition in both a prophylaxis and treatment mode. And based in part on these results, this drug is now in clinical trials across 20 sites in Africa for COVID-19. More recently, we were doing studies with influenza on our alveolus chip. And we do these often with or without breathing motions to see if there's anything interesting. And what we found is that physiological breathing motions suppressed viral infection dramatically, as well as the inflammatory cytokine production. And what we found, long story short, published in Nature Communications a few months ago, is that breathing activates innate immunity, the protective type 1 and 3 interferons that are our first line of defense against viral infection. Also, it could explain in part the tumor effects that we found, because interferons can protect against tumor growth as well. Now, when we did RNA-seq, and what we found with plus or minus breathing, one gene that caught our eye was this S100A7, which is a ligand for the RAGE receptor, the receptor for advanced glycation end products, which is actually most highly expressed in alveolar epithelial cells and is a major mediator of inflammation around the body. And so we identified a drug called aziluricin that's a RAGE inhibitor, and we found that we can completely suppress the cytokine storm, in this case induced by influenza H3N2, using this drug. And based on this findings, a drug company called Cantex licensed our intellectual property on this, and they included these very data in an IND application to the FDA that's currently being considered to move this drug to COVID-19 trials. Now, we've also used this for discovery. We carried out a genome-wide CRISPR screen that basically led us to identify a series of short duplex RNAs that we thought were acting like siRNAs, but in fact, they were acting not by targeting a gene, but they all had the same unique structure. This is also published recently. They're 20 base pairs, and they are potent inducers of type 1 and 3 interferons. And here you see interferon beta, 30-fold increase, almost 15-fold increase in alveolar and airway human epithelium. And now we found that these are potent inhibitors, basically generic inhibitors of many different types of viral infection. This is inhibiting H1N1 influenza, H3N2 influenza, SARS-CoV-2, 5 log reduction in vitro, MERS, and NL63 is another coronavirus that causes the common cold. And more impressively, we carried out studies in an animal model of SARS-CoV-2 in a BSL-3 lab, and we confirm a 5 log reduction in the lungs of these animals. And we're trying to move this towards the clinic as well. Now, we've made many different organ chips. So lung was where we started. We then created a chip model of intestine. We started with human CACO2 intestinal epithelial cells, but now we use primary patient-derived organoids as a source. I'm sure you're all familiar with organoids. They have stem cells. They retain cell differentiation capabilities, usually grown in a 3D gel. But it's really not an organoid. It's really a tissueoid. And so we break it up and then put them on the top of our chip with endothelial cells on the bottom. And now you can get beautiful intestinal villi, but now you can measure absorption, put microbiome, look at mucus production, et cetera. Now, the first thing we did is transcriptomics. And just to show you here, the right lane is transcriptomics of genes involved in many relevant pathways in the duodenum from in vivo. The left lane is an organoid created from the duodenum in vitro in a 3D gel. And the middle lane is our organ chips created with those cells from the organoid. And you can see we're getting much closer to in vivo using the organ chip model. I should note that, this is literally giving peristaltic-like motions plus trickling flow, and even CACO2 cells that grow flat in a dish form intestinal villi under these conditions. We also used organoids from colon, and this is a cross-section of the chip showing you the fidelity of mimicry of tissue architecture we get with polarized epithelium, magenta-filled goblet cells, apical, microvilli, et cetera. And now one of the nice tricks here is that we can look at mucus production by turning the chip sideways and using dark field imaging. And this white fuzz is mucus, which we can watch and can accumulate in real time in living cultures just by turning the chip on its side. And for the first time, we were able to show that we can recapitulate the bilayer structure and thickness of human colonic mucus in vitro, which has a permeable and non-penetrable layer that is what protects us against the microbiome and pathogens. Now, because of this, we explored whether we can actually co-culture complex living microbiome in contact with human cells for extended times. Now, people try to do this in organoids. They take a needle, they inject bugs in the lumen, and for 16 to 24 hours, they can study it, and then it dies. But what we've done is we've created a hypoxia gradient across the chip so that we can look at both aerobes and obligate anaerobes. We have oxygen sensors in line, so we can confirm we can get below 0.5% oxygen doing this. And now we can culture literally a stool sample, take it, dissolve it, put it on chip, and for 5 days in culture, we actually can retain over 200 unique operational taxonomic units, which is basically strains and species of bacteria from 11 different genera, with a similar complexity of what you see in human stool, both anaerobes and aerobes. At the right is 3 days, a living video of showing the density of microbes from a patient in the neonatal intensive care unit, a stool specimen, and these are the villi, and it's just filled with bacteria. And we actually, if they're a healthy microbiome, we get even better barrier function, which is quite amazing. Now, having done this, Gates Foundation came to us and asked if we could develop a model of environmental enteric dysfunction, EED, which is malnutrition in kids in the low resource nations. And the contributors to that are malnutrition, dysbiosis, abnormal microbiome, pathogen, immune response, and we can control each of these individually. So what we were able to do is we were able to obtain organoids from the duodenum of kids in Pakistan who have EED, and we compared their organoid-based cells on chip with healthy organoid-based cells from similar age kids in the presence or absence of nutritional deficiency, which is minus nicotinamide and tryptophan, minus N minus T. The first thing you see is you replicate the villus blunting seen in these patients. And when we did transcriptomic analysis under these conditions, what we saw is that we were lucky to have transcriptomics of the duodenum of patients, shown at the left. And you can see that if we have just compare the chips, control chips at the right lane from patients versus healthy with full medium, doesn't look anything like it. The reds are high at the bottom and red, as well as at the top. If you have a healthy chip with nutritional deficiency, it doesn't look like it. But if you have a EED chip with nutritional deficiency, now it's beginning to get blue at the bottom and more red at the top, and we're approaching the clinical phenotype. And we also quantified absorption of nutrients, fatty acids, proteins, and we mimic the clinical phenotype as well. And more recently, we've added the next layer of complexity because we collaborated with Jeff Gordon, who actually has isolates of microbiome from these patients. And now when we put them on chip and look at multiple cytokines, you can see that when you have the healthy microbiome on these chips versus the EED microbiome, we get much higher levels of various inflammatory cytokines. And we also can get synergy or additive effects in terms of disruption of the intestinal barrier and intestinal permeability. I mean, I show you this just to show you again how you can control all these parameters that are relevant for clinical phenotype that you really can't do easily in vitro otherwise or in animals. Based on that, Gates asked us to then develop a human vagina chip, which was just published about a week ago, where now we have human vaginal epithelium on top, human vaginal fibroblasts on the bottom to have epithelial stromal interface. And you can see we get normal stratified epithelial differentiation. But the reason they wanted us to do this is they're trying to move a live biotherapeutic product, which is a consortia of healthy L. crispatus bacteria into clinical trials for bacterial vaginosis in third world, low resource nations. So bacterial vaginosis, is actually a major cause of prenatal labor and preterm birth and death. And so the healthy vagina is dominated by L. crispatus consortia, whereas bacterial vaginosis, it flips to a state that's dominated by more pathogenic bacteria like Gardnerella vaginalis. So in this study, we tested 4 different formulations of consortia that the Gates Foundation is working on. And you can see that just compared to baseline, we suppress inflammatory cytokine production by this L. crispatus, potential therapeutic consortium. And then more recently, we tested this against, first of all, we could show that if we incubate Gardnerella vaginalis, Gardnerella, which is the bad bug, we can actually see in black bars increase in various cytokines. But now if we treat them with this biotherapeutic product that is supposed to be therapeutic, we actually decrease cytokine production in these conditions. And so this actually is moving to clinical trials in Africa in the near future. The other thing that we began to do is really to explore the effect of the stromal environment on epithelial phenotype and diseases. So here we got colon organoids from patients with inflammatory bowel disease versus healthy, as well as fibroblasts from those same patients. And we can now do tissue recombinants in vitro. Very interestingly, we found that the inflammatory fibroblasts have a major contributor to this IBD phenotype. For example, if you take a healthy epithelium with an inflammatory fibroblast, you have increased intestinal permeability, decreased barrier function, and if you combine it with the normal fibroblasts, you don't see that. If you now also look at inflammatory cytokine production, you can see, look at this increase on the top right with the fibroblasts in healthy epithelium or IBD, you get increased IL-6, but greatly pronounced in the IBD. So there's a synergy here. But even more interestingly, if you add the IBD fibroblasts, epithelium with true normal fibroblasts, you can suppress this IL-6. So the epithelium stroma interactions are playing a key role in this disease, which is a precancerous condition. Whereas at the bottom, IL-8, you see the ability to increase in the healthy, but now the normal does not have a protective effect. So we're just beginning to explore this. But one thing we've done recently is we added carcinogens into this model, and we could actually see changes in mutation that are associated with cancer progression. So very interesting way to begin to study much more complex processes. With funding from a Storming Cancer grant, we've also been looking at the effects of the stromal microenvironment in chronic inflammation-associated cancer, specifically esophageal cancer, where we're getting from Lorenzo Ferri's lab in McGill. He's a surgeon, and he gets from the same patient, we can get, he takes out the entire esophagus when they have cancer, and there are regions that are cancerous or dysplastic, other regions that are Barrett's metaplasia, which is precancerous, as well as adjacent normal. And then we also can get samples from, cadavers from normal esophagus. And the idea is to put these on chips and look at, we're getting the stroma as well as the epithelium, and look at stromal epithelial interplay. And this is something we just submitted, which is if you take healthy esophageal epithelium and healthy fibroblasts and you put them on chip together, you get a stratified epithelium like in vivo. But if you take the metaplastic fibroblasts from Barrett's and the metaplastic epithelium, you get this disorganized looking structure that looks nothing like a stratified epithelium, more like what we see in metaplasia. And when you stain for multiple markers of Barrett's metaplasia, lefty 1 is a classic TFF3. It looks just like in vivo. The bottom is a histological section from one of these patients. And now when we do the stromal mixes or recombinations, what we can see is that if you have stroma from dysplasia or cancerous regions and you mix it with the metaplastic, you get a significant increase in growth measured by Ki-67. You see it here by labeling. But interestingly, if you have the adjacent normal fibroblasts, not only do you have lower growth, but you begin to get a little bit more organization in terms of basal nuclei, polarized epithelium, more regularity than you do in these other conditions. And so this is just beginning, but you actually can use this model to study epithelial stromal interactions. Now, before I end, there are some other key goals in this field, and I think for personalized medicine that are extremely relevant. One is to predict human drug responses using clinically relevant dose exposures. Usually studies in vitro, you bathe cells in drug at high dose for longer times than it's given to a patient, and in a patient, obviously, drugs usually go up and down depending on dose regimen due to differences in pharmacokinetics. That's almost always ignored in vitro. The other is to develop personalized model for individual patients, to create models that replicate complex immune responses, which is so important now with immuno-oncology and other areas. And also, as I show you, to recapitulate species-specific responses. So we published a couple of years ago a human bone marrow chip where we take CD34 cells from either blood or marrow from patients, individual patients. We then put them in a matrix gel with bone marrow-derived stromal cells. We put endothelial cells on the other side, and we only perfuse through the vascular channel. And over a month in culture, we get near normal neutrophil maturation and erythroid maturation as confirmed by FACS sorting. Now, I'm funded by FDA to look at radiation toxicity. And the first thing we did is we could show that not only we do show radiation toxicity, but we show the same dose sensitivity about 1 to 2 gray that's seen in patients, where this is not mimicked in animals. Each colored dot is a different patient-derived chip showing you the robustness and in terms of replicas from patient to patient variability. Actually, is pretty minimal here. We then tested dose responsivity to a cancer drug, 5-FU, 5-fluorouracil, which is known to induce marrow toxicity. In patients, they receive a dose at about 4 micromolar for 2 days IV. But when people have shown that this is toxic in vitro, they've done 5 days with high dose. When we do it in our chips, we can show, as shown by highlighted by the colored bar, we get toxicity at 4 micromolar that you do not see in suspension culture or even in the same gel under static conditions. And again, you can see that patient-patient variability is quite good. Now, more interestingly, AstraZeneca, a pharmaceutical company, came to us because they had a drug in phase 1 clinical trials that had a very peculiar regimen-specific toxicity. The same dose over 2 hours or 48 hours gave, in 2 hours, you saw neutropenia and anemia, but in 8 hours, only neutropenia, and they couldn't model this in animals. And luckily, they had done the pharmacokinetics analysis and measured by mass spec drug levels in patients, which are shown in gray in the left graphs. Now, because we have flow, we could recapitulate precisely the PK of this drug on chip, and those are the open circles in black. When we do that, we recapitulate this regimen-specific toxicity with neutropenia and anemia, or red blood cell and white blood cell decreases in the top, where primarily neutrophil at the bottom. And this was not seen in static culture. Now, even more interestingly, we could wash the drug out and see recovery, and that could not even be seen in any models they had. And that's obviously extremely relevant for drug development and deciding on what to give the patient. Now, using this, we also pursued personalized models. So we collaborated with Kiko Shimomura and Cole Novena at Boston Children's and Dana-Farber. They were interested in a rare genetic disorder called, oh my God, I don't have it. Oh, it's over here. Shwachman-Diamond syndrome, or SDS, which is a rare genetic disorder that leads to abnormal blood cell formation. And what you see in the bottom left is viewing the whole chip at low mag. There's just less cellularity in these chips, SDS chips. We got CD34 cells from these patients. We found quantified reduction in neutrophil, erythroid, and CD34 cells. But what was interesting is when we did FACS sorting looking at CD13 and 16, we observed a neutrophil maturation defect that has never been seen before or described before in these patients. And we then went back to the archives and we were able to look at 8 patients because we only done 2. And we found that indeed there are 2 patient populations, one of which has low CD13 levels and one which does not. And so this shows an example of getting insight into individual patient response. And I should note that Lorenzo Ferri, who provided those esophageal cancer cells, cancer and metaplasia cell for our studies, is now using chips lined with patient-derived esophageal cancer cells and testing different drug regimens that he's considering to try to optimize it for the patients. I showed this earlier. There was this cover of the New York Times Sunday section a couple of years ago where it basically said future vaccine depend on test subjects and short supply monkeys. And this is a real problem using NHPs for vaccine development and other biologic development. And so wouldn't it be nice if you could test these vaccines and drugs in vitro? And we have now done this. We published early this year a human lymphoid follicle chip, which is amazingly simple. We take B and T cells from blood, put them at high density in a matrix gel in one channel, just perfuse through the other channel. And what we see is that they spontaneously aggregate into small groupings that, when you provide an antigen, form germinal center-like structures with CTLA-4 cells, as well as T and B cells at high density, plasma cells, cytokines are produced like CXCL13. We can get, if we vaccinate with a commercial flu zone vaccine, influenza vaccine, we get this entire response, including production of high affinity anti-influenza hemagglutinin IgG. I should say we have dendritic cells here as well. We could also collect the outflow from the vascular channel, and we can measure cytokine production. And luckily, we had measurements from patients who were immunized for influenza, and we could show very similar profiles of interferon gamma, IL-10, IL-2, GM-CSF, et cetera, in our chips as in patients. And we could also demonstrate this is very good for quantifying adjuvants. We've now been working with multiple drug companies and Gates Foundation on mRNA vaccines, protein vaccines, replicating vaccines. We've even been modeling Lyme disease now on these chips, which is quite amazing. And that study was with FluZone, was a memory response because we're all seeing flu, but here we can confirm we can get a novice response using rabies vaccine and IgG production. And we are also linking this, the lymphoid follicle chip, to other organ chips to look at more complex immune responses. For example, we've linked it to the intestine chip and looked at mucosal, infected with polio vaccine, and a vaccine, and look at mucosal vaccination and can see a vaccination response. Now, I mentioned species-specific chips, and this was not obvious, but early on, two pharmaceutical companies, AstraZeneca and Janssen, came to me and said, will you make dog and rat liver chips? While we were already doing human. And they pointed out that FDA requires animal studies for drug-induced liver toxicity injury, drug-induced liver injury, and almost always it's dog and rat or a rodent, and they almost always get conflicting results, and therefore they have to make decisions that are based, on gut feeling, and that often leads to failure. So in this study published a few years ago, we made human, dog, and rat chips. They have 4 different cell types: primary species-specific hepatocytes, liver sinusoidal endothelial cells, Kupffer cells, and stellate cells in their correct positions. And here you're seeing one result with bosentan, where we beautifully mimic the species-specific toxicities. And in this paper, we recapitulated not only hepatocellular injury, but steatosis, cholestasis, and fibrosis. Now, last Tuesday, there was a paper published by Emulate, which is a company I founded that is commercializing this technology. I'm part of it as well, but it was really done there, where they carried out studies with 870 human liver chips from 3 different patient donors. They tested a blinded set of 27 drugs that are known to be hepatotoxic or non-toxic. They were identified, most of them identified by a pharmaceutical consortium called the IQ Consortium, where they published a paper saying, we know the results in animals, we know the results in humans, they're often completely different. If you could do better than this, this really is what we're looking for. And the net sum is that in this study, they could show that these human liver chips predicted drug-induced liver injury with 87% sensitivity and 100% specificity. And this was 7 to 8 times better than animal models. And they did an economic analysis, and this one replacement of animal models in this, just for one, context of use, drug-induced liver injury, they estimate could save the pharmaceutical industry $3 billion a year in terms of lost dollars because of later failures in clinical trials. And I really hope this will represent a big change. So to pull this together, we've created models of probably 15 different organ chips, and at the Wyss Institute, other groups have done even more. But when I first presented this, I had a review where I said that we might be able to create an integrated human body on chips because we have a endothelium line vascular channel. We should be able to transfer flow from one channel to the other. So you can imagine an oral drug through the gut chip, watch it be absorbed, see it metabolized by the liver chip, peed out by the kidney chip. What does it do to heart toxin? How does it work on bone? And believe it or not, we've done this. We published a few years ago. We don't use tubes to connect chips. We use a robotic sampler, so we can do drop by drop from chip to chip in any order, and then take a drop for mass spec for drug levels or do cytokine analysis. And in this study, we had gut, liver, kidney, heart, lung, blood-brain barrier, brain, skin, bone marrow, and pancreas. And this instrument was able to keep them alive and image them for a month while interconnected. But the real reason we were funded to do this by DARPA was actually to develop a way to carry out in vitro to in vivo translation of drug PK parameters. So in other words, could we predict drug pharmacokinetics or optimize them in vitro before we go to a clinical trial, and thus design a clinical trial and shortcut, go right to the regimen you're looking at. And to do this, we simplified. We made a first pass model with a gut, liver, kidney, and we integrated what we call an arteriovenous reservoir so that you're not collecting a sample right after any one chip, but they're mixed just like taking a peripheral blood. And we collaborate with a company called CFDRC that does computational PKPD for pharma right now with in vivo samples. But we're able to use their computation models to scale up from our little chip to real organs based on known volumes, tissue masses, blood flows, et cetera. And what you want to be able to do is predict key parameters like the Cmax and T1/2. And so what you see here is published 2 years ago. We did 2 drugs, oral nicotine, and we put it through the lumen of the gut chip. We used mass spec to quantify its levels that it was absorbed, levels passed to the liver chip, whether it was metabolized, levels in the kidney chip and peed out, and then finally the level in the reservoir. We did the same with IV cisplatin. Because it was IV, we didn't need the gut chip, so we put a bone marrow chip to look at pharmacodynamics because that's where it has toxicity. And at the right are the results. The dotted lines are predictions of our computational model, same model we use for both. The dots with error bars are from past clinical studies. In the top, it was from Sweden. They called chewing tobacco snus. So it was nicotine in 2 different formulations of chewing tobacco versus gum at 3 different doses. And we precisely predict the PK. And the same with cisplatin, 2 different doses IV infusion from a past clinical study. So to end, I think these 2-channel organ chips can faithfully recapitulate human pathophysiology. They mimic human responses to drugs and radiation using clinically relevant dose exposures. They could quantitatively predict drug PK. And I think we believe that these are really in a place now that they can replace some animal models, both in research labs and in the drug development pipeline, and be used for personalized medicine. And, I think I had a review years ago, where it really emphasized this idea of synthetic biology at the cell, tissue, and organ level. If you use just the epithelium and you mimic an in vivo response, maybe there's not as much complex regulation as you thought. But if you can't, something's missing. And now you could add back endothelium and stroma and immune cells and microbiome, one at a time or in combination, and tease it out. So you can get insight into disease mechanism as well as drug mechanism of action and drug mechanism of toxicity. And obviously, major implications for personalized medicine with patient-derived cells, iPS cells, organoids. You can make single chips, multi-organ chips. You can, as I showed you, carry out modeling of rare genetic disorders when it's hard to get many patients together. You could carry out toxicity testing where it's unethical in humans, like lethal radiation or prenatal testing in pregnant women. You can compare drug responses in male versus female, which we're beginning to do, or old versus young. But I think the real game changer here is changing the way clinical trials are carried out. Right now, big pharma will spend tens of millions or hundreds of millions on drug development, tens of millions on a clinical trial, and they almost always fail. Then they'll do number crunching with statisticians to see if there's a genetic subgroup that might have responded better. If they're lucky, they find it, they'll do a small targeted trial and get approved for narrow application. with organs on chips and organoids, iPS cells, patient-derived cells, you can imagine identifying a small subgroup of patients who are very similar genetically or other phenotypically, and take 50 Hispanic women who have asthma, ultra sensitive to cigarette smoke, make chips for those women, identify the best drug out there in terms of efficacy, and then test other organs so that there's minimizing toxicity, and then do a small trial with those 50 women. I think that would in fact, have safer drugs that are more effective at lower cost and much, much faster. So with that, as a Harvard faculty, I must disclose that you can't believe anything I say, because I'm a member of the board, chair the SAB, and hold equity in Emulate, which is now distributing automated systems to run the chips and the chips themselves around the world. And I could not do this without an incredibly multidisciplinary team at the Wyss Institute. We have expertise in every area and incredible prototyping capabilities. And with that, I invite you to our website. I think you will enjoy it. There's amazing stuff going on. So I'm happy to answer questions. Thank you.
Great. Thanks very much, Don. It's I really don't know that I've seen anything quite like that in trying to replicate human tissues or really organs ex vivo in that way. I have a couple questions. I'll just remind other people, if you have questions, please put up your hands or enter them in the chat and I'll refer them to Don. So one thing I was wondering, going all the way back to your studies of viral infection in the lung, you tried a bunch of drugs and found that there were effects on, for instance, cytokine secretion. One of the things I was wondering is, did you ever try corticosteroids? And if they had an impact, was it immune cell dependent?
I can't remember. I kind of vaguely remember that we were going to it. I don't think we did. We were funded. It was COVID emergency, and we were screening through. We were using computational approaches on hundreds of drugs, but I don't think we did at the time, because we were looking at initial. We only had pseudotype virus, so we couldn't really do something like dexamethasone, which is going to be involved in the later response. We could look at cytokine production, but I don't think we did. If I'll ask, if we do, I'll let you know separately.
And the reason why I ask is that it is probably the most wide for people who are hospitalized with COVID.
Yeah.
Generally for hypoxemia, it is the most commonly used drug. And I would have to say the mechanism of action really isn't that clearly understood. And it would be interesting to see if it was immune cell dependent or immune cell independent. You probably have the capacity to answer that.
Yeah, no, that's a good point. I mean, we've integrated macrophages into these chips, which have a major effect on viral infection and other immune cells as well. And so we should go back and look at that. It's a good point.
Also, you did some studies, I think, both in the IBD as well as the colon cancer context. I think it was colon cancer. Anyway, a couple contexts where you saw these disease-associated fibroblasts, whether they're IBD-associated fibroblasts or cancer-associated fibroblasts, were by themselves exerting a function or exerting a phenotype sort of on even healthy normal cells. And I was wondering, have you studied like the half-life of that? I mean, one might assume that these fibroblasts aren't genetically altered, but they're rather somehow, non-genetically imprinted in a way to behaving the way they are. And is that a reversible phenotype so that you could reverse, after a certain amount of, say you have this CAFs away from the cancer or the IBD fibroblasts away from the IBD, do they return to a normal phenotype in time?
That's a great question. I mean, the one thing that has struck home for me is that the importance of epigenetics, because we take COPD patient cells without immune cells and they retain the phenotype. Every time we take adult cells, they retain the phenotype. Those fibroblasts are cultured multiple times in vitro and they seem to retain the phenotype. But I think targeting epigenetics, like can you reverse that, is a great goal and the type of thing that we could do with these systems. But we haven't quantified time in a quantitative way.
Yeah, that'd be really cool to see.
It's a really good. It's actually a great idea. Yeah, because I've always said that like we say it's epigenetic, but there are therapies out there people are developing to target those, epigenetic modifications. So we really should go back and look at that.
Right. And even if they're sort of blunt tool epigenetic drugs,
Right.
that might be better than, reshuffling the deck.
Yeah.
Might
But
be.
the
might be
nice
better.
thing is we could test each cell type individually or together. Do you know what I mean? Like we could pre-treat the fibroblasts and see if, we can really reverse that, or we treat them together. There may be interplay between the two. I see a question that just came up.
Yeah, I'll read it out. It says, it's from Lindsay. Can the chips be used? Can the chips be used to model resistance in cancer chemotherapy and targeted therapeutics? Do you have, is the time long enough to model that?
Given what we found with viral resistance, I would think not only could you see whether there's initial resistance, but you could see whether there's a propensity to become resistant quickly in these model systems, like with specific patients. I think I haven't seen the results yet, but the collaborator at McGill is using patient-derived esophageal cancer cells on chip. He definitely is seeing differences in sensitivity to the same drugs in a patient-specific way. But we haven't done it, but it should be approachable.
So this, I'll just ask this, we'll make this the final question. This brings up the question in my mind, like, how long do these things last? You establish a primary culture or organ culture in one of these things. How long is it sitting around viable?
I mean, usually a month is, very doable, depending on the system, but it takes a few weeks to differentiate them before you start your experiment, but we usually do weeks to a month afterwards. Some can go longer. It may be possible to go longer if you're absolutely, trying to do that, but we haven't pursued that.
Oh, cool. Well, thanks a lot. That's for a really unique and eye-opening talk, Don. I really appreciate you sharing with the society.
My pleasure. Thanks. Bye-bye.
All right. Bye, everyone.